Hello, my name is Dr. Michael Granatosky, and I'm an assistant professor in the anatomy department here at NYITCOM. I'm also the robotics group leader at the Center for Biomedical Innovation. I would love to be there with all of you right now at OMED, but in lieu of that, I'm excited to show you some of the research we've been doing in the Comparative Animal Motion Laboratory. So we're a biomechanics lab, and our bread and butter involves really trying to understand the underlying mechanisms that drive animal performance. We use a combination of both ex vivo and in vivo experiments. If you look in the black box over there, you'll see some of the work we've been using, taking high-resolution micro-CT scans and combining them with DICE CT data to actually model out the individual muscle fascicles. What this allows us to do is it allows us to no longer think of muscles like the temporalis just as one muscle, but instead get into the actual function of these very, very tiny little fascicles on a small micro scale. If you look at the other screen, you'll actually see some of our live animal work, where we're doing a project focusing on the bipedal locomotion of parrots. You'll also notice on the screen there are these little tiny dots that might be jiggling just a little bit. What these are is these are actually kinematic markers that have been mapped out using machine learning. What this does is this takes the slide-by-slide presentation of actually manually putting where those markers are and reduces the time considerably. With this speed in data analysis, this means we can pump out a lot more data and really excitingly actually bring this out in the field, away from the clinic, away from the laboratories. And this kind of work is actually the focus of one of the NYITCOM DO PH.D. students we have here. So beyond just understanding what these animals are doing, my lab also wants to take advantage of the fact that natural selection's had millions of years to tinker and experiment with certain designs that have made animals ideally adapted for their environment. And so what we're doing is we're learning from these animals to actually develop bio-inspired robotics. So we have a streamlined process for trying to collect and process all this data. All of it starts with very, very high resolution kinematic tracking, like you're watching here on this treadmill. Afterwards, we're able to actually go a step further using a technique called XROM, or X-ray reconstruction of moving morphology. This allows us to actually watch how the animal's moving using X-ray cameras. And from there, we can actually superimpose high-resolution CT scans of the bones themselves. Here at NYITCOM, we can go a step further by actually taking robotic structures and imposing them onto these high-resolution CT scans. From there, we can move out of biological space and get into robotic space. Now, one of the projects that we're most excited about is actually trying to understand climbing and climbing systems. It turns out to be the case that one of the leading causes of mortality in terms of workplace injuries are actually falls from heights. Well, many animals have figured out how to move in trees fluid and quickly. And so we want to see if we could take advantage of that to develop robotic systems that are capable of climbing for us. What you're seeing here is actually a climbing woodpecker. And while there are many aspects of its anatomy that allow it to do this remarkable vertical locomotion, what I'm going to have you focus on here today is the actual tail. You'll see what this animal's doing is it's acting like a strut to prevent itself from toppling backwards. Well, we've had students here at NYITCOM actually develop these models to make these three-dimensional movements of those robotic parts. So it's actually able to serve as sort of this passive strut mechanism in our robotic system. And we're excited about developing this research further in the next coming months. Well, I really just also want to give a shout out to all the students in my lab. None of this work could be done without them. And I really appreciate your time and hope you're enjoying your OMED conference.